red reported virus, by far the biggest pest species as fire ants go. But in addition, there's also a black fire ant called fish fry that has been introduced into the US. So both of these are from South America. Fish fry is a little bit more temperate tolerance. This is the current distribution of invasive fire ants in the US. So in the 19-teens, Sonox's fish fry was actually first introduced into Mobile, Alabama. They kind of hung out around Mobile until Invicta was then introduced in the 1930s. And when that happened, Invicta started to spread, and then really that spread happened in the 50s and 60s to the current distribution that we have now, in addition to multiple other places around the world. And so fish fry has really been fairly geographically restricted and gone pretty much northward from Mobile, Alabama, while Invicta really has spread far and wide. So in their native ranges, these two species, while they are parapatric for part of their ranges, we have no evidence of gene flow or interaction between them in their native ranges. However, in 1985, using biochemical analyses, it was first discovered that in the US, these two species are actually hybridizing. And then people started looking at museum specimens and realized that when Invicta got introduced in the 30s, hybridization already had started occurring in the US between those two species. So this hybrid zone, so fish fry is relegated towards northern Mississippi and Tennessee. Oh my goodness. So while Invicta is everywhere else. And this hybrid zone has been formed between the two of them. And so as these two species have migrated and invaded the south, this hybrid zone has also migrated and invaded. So we know quite a lot about this hybrid zone. We know that it's shifted northward and expanded. We know that it doesn't play by the rules of a lot of other hybrid zones, because once again, it's only less than 100 years old between two species that have been previously had no integration between them. We know it's a very large and wide hybrid zone, and that even in this one corner where potential differential species could be found co-occurring in the same geographic region, we don't find them. So if you find Invicta, you'll never find a colony of fish fry right next to it, but you will find hybrids with either one of the potential species. And we also know that this hybrid zone doesn't play by the normal rules of tension zone, and it's more of a mosaic zone. And it's partly because it's such a young zone, partly because it's been pushed by invasive species that are colonizing new habitats, and partly because as humans, we're still distributing these things and moving them around in South quite a lot. So it's just as likely that you're moving sod from southern Mississippi with Invicta into it up into north Mississippi, where fish fry is actually present. In previous studies, we also know that in the hybrids, they're more likely to have Invicta mitochondrial haplotypes. So previous genetic studies of this have mainly been survey studies to try and figure out how this hybrid zone is moving throughout the landscape. They've been focused on a few molecular markers, the FLPs, a handful of microsatellites. And because they're surveys, most of them have relatively smaller population sizes. They've also been focused on monogyne colonies. So fire ants are cool for a lot of reasons, and this is one of them. The fish fry and Invicta are a subgroup of fire ants that have two social forms, both monogyne and polygyne. And so if we focus on monogyne colonies are formed by one relatively large female. She goes on a mating flight, mates, and then she goes and sets up her own colony. So all of her offspring are in that colony. They're all highly related to each other. Her fitness is relatively high, and she can actually survive and found a colony. Alternatively, we have another social form called polygyne. And so in a polygyne colony, you can have 3 to hundreds of queens. And these have very different life history traits. So for instance, their individual queens are relatively smaller. Many are unmated. So in ants, we have haplodiploidy, where females are diploid, and males are haploid. So while these unmated females can't produce workers or daughter queens, they can still produce sons. And then oftentimes colony founding is by budding. So you take a few workers from the maternal colony and just walk a little ways away from it and start a new colony, or these queens just try and invade another colony and get incorporated into it. And so the aggression level between monogyne and polygynes are quite different, where polygynes can form super colonies throughout a large area where ants can just mix between nests. And individuals kind of being in fitness per queen is much lower in polygyne populations than in monogyne populations. And interestingly enough, this is all tied to one Mendelian locus called GP9, where monogynes are homozygous for the dominant allele, and polygyne queens have to be heterozygous for this allele. So my study is really looking at how much genetic diversity can we find in these hybrid populations, given what we already know about it being mosaic, hybrid zone, and can we detect different levels of hybridization between these monogyne and polygyne populations in admixture and also in mitochondrial and nuclear. So to do this, rather than do survey studies, we focused on a few populations and we took a lot of individuals from each one. So we have one pure Richteri population, we have 4 pure Invicta populations, one of which is polygyne, and then we have 3 populations of hybrids, one of which is also polygyne. So another factor to this is both Richteri and Invicta in their South American ranges have both social forms, but in the US, only Invicta has both social forms. So we know that in this hybrid polygyne population, what makes those polygyne have to be coming from the Invicta, not the Richteri individuals. So for each of these populations, we took one alate, so a female winged daughter, from the colony and sequenced CO1 and 66 microsatellite loci, 24 of them which were species diagnostic. And these mapped to 14 out of the 16 chromosomes of the Invicta genome. We also took workers from each of the nests and made sure that they were in fact polygyne and monogyne. So when I analyzed my microsatellite data, this is just using a simple PCA plot, you can clearly see, very nicely, that Richteri and Invicta are clearly differentiated by our markers, and that our hybrids are really dispersed quite in between the two. So these are colored, each individual is colored by what population they come from. So when we look at structure, we have a beautiful structure plots. So all 4 of our Invicta pure parental populations, regardless if they're monogyne or polygyne, are clearly Invicta. Our Richteri parental populations were a little bit more interesting. So I divided it into 2, and that's because this population over here is, we went to this population in Tennessee and sampled it in 2005 and 2007. We sampled it again in 2011. The ones from 2005 and 2007 are pure. All the same mitochondrial haplotype, clearly Richteri, and in 2011, a few of these individuals have Invicta mitochondrial haplotypes, and at least 2 are clearly hybrids. So this map is drawn based on survey data in the late 2000s, and we think that this hybrid zone has even moved much farther since then. If you look at our hybrids, our Tennessee population, which is by far the farthest from either of the parental populations, is actually the most differentiated between all of the hybrid populations. So a monogyne population is more different than a monogyne versus a polygyne population, which are both found here in Mississippi, which is probably not surprising given the geographic distance between these and the fact that the Tennessee population is so far from either of the parental species. So next I looked at this data by using the program View Hybrids, and this is a program that I use with only the 24 diagnostic loci, and it bins each individual into genetic categories of if they're a pure individual, backcrossed F1, F2, that sort of thing. And then I've divided individuals between monogyne and polygyne populations, and you can see here their hybrid index that I calculated with intergress. And so one of the things, one of the most surprising findings is that not one individual in any of these populations was deemed to be an F1, which is perhaps not surprising since we don't ever find a Richteri and Invicta population right next to each other. What is surprising about this is that almost everything is considered to be an F2, and I think that this is a limitation of the program that they're not actually F2s, they're hybrids of multiple generations. The other interesting thing is there are very few backcross individuals, even in populations where we know we have hybrids right next to Invicta or hybrids right next to fish dry. Very, very few colonies actually have the backcross. In addition, I looked at this using ancient Rust. So once again, only using the 24 diagnostic markers and training the model based just on the frontals. So in this plot, you have your hybrid index on this scale, and then your intraspecific heterozygosity. So if you're 0, you're homozygous at every locus for one species alleles versus another, and at 1, you're heterozygous at every single locus for Restrine and Invicta. And so, once again, we find no F1s, very few backcrossed individuals, and a really large spread of genetic diversity. And this is really, we think hybrids are preferentially mating with hybrids and aren't quite fixed. So next we sequenced CO1 and found a mitochondrial phylogeny, which is similar to what has been found before, that there are multiple Invicta mitochondrial haplotypes. So all of these ones named H, we can actually trace back to where they originated in South America. And these are the haplotypes that we know to be present in the US. And then there's only one Restrine mitochondrial haplotype in the US. So if we look at the prevalence of these haplotypes in our populations, we can see our pure Invicta populations only have Invicta mitochondrial haplotypes, which are color-coded up here. Our 2005-2007 Restrine population only has Restrine, with a few Invicta mitochondrial haplotypes in the 2011 section. And then our, consistent with previous reports, in our 2 monogyne hybrid populations, we only find about a quarter of the individuals have a Restrine mitochondrial haplotype. But surprisingly, in our 1 polygyne population, we have 50% Restrine mitochondrial haplotypes. So then I looked at this, once again, dividing it between our 3 hybrid populations and by our hybrid index. And so each individual is color-coded by the mitochondrial haplotype. And I wanted to know if certain mitochondrial haplotypes were associated with certain genomic arrangements or hybrid indexes. And so that doesn't seem to be the case at all. All the mitochondrial haplotypes are fully distributed across the index. The only exception to that is actually, once again, our Tennessee population, which is a monogyne population, and it seems to be shifted more towards Restrine-like hybrid index in general. So overall, we've learned that our hybrids are pretty much never F1s, that we have very few backcrossed colonies and a larger genetic variation in our French populations. And we think this is really because hybrids are preferentially mating with hybrids, regardless of what social form they take on. We also found that there was no real difference in gene flow and admixture between our monogyne and our polygyne colonies. And then, pretty surprisingly, we see asymmetric gene flow in our monogyne, but not in our polygyne colonies.